To get started with our first talk of the afternoon before we head into our lab sessions, I'm very excited to introduce Charlotte Bunne, who is an assistant professor at EPFL in the Computer Science and Life Sciences Department. Before that, she was a postdoc at Genentech in Stanford, and before that, she completed a PhD in computer science at ETH Zurich, working with Andreas Krauss and Marco Cuderi. During her graduate studies, she was also a visiting researcher at the Broad Institute of MIT and Harvard, hosted by Ann Carpenter, who you heard from this morning, and Shantanu Singh, and worked with Stephanie Jekla at MIT. Her research aims to advance personalized medicine by utilizing machine learning and large-scale biomedical data. I'm very excited to welcome Charlotte, who is tuning in remotely today. So if we can all, again, turn back to the camera and wave hi to Charlotte so she can see us. Perfect. And I'll hand it over to Charlotte. Thanks, Charlotte.
Hey, thanks a lot for having me. And I'm very sorry I couldn't be here today in person. I'm somewhat restricted in the type of travel I can do currently. So I'm going to speak about how to use generative modeling. And this is a very broad term, in particular, diffusion models and neural optimal transport to enhance certain aspects of personalized medicine. So let's dive right in. As Johnny already introduced, so I just finished my PhD and I did a very short interim postdoc before now starting at EPFL. And so there I'm going to lead the AI in molecular medicine lab. In case the research I'm going to present today is something that interests you and inspires you and you're still looking for research opportunities, please reach out. Our lab homepage is already launched. And if you have questions about EPFL, how to apply, there's also a future PhD student who's joining my lab, Ishan, so you can ask him all about how it is to apply at EPFL and what life at EPFL looks like. So let's dive right in. We are going to cover quite some things with like two somewhat more theory-heavy parts, but the rest is relatively light. So first, I'm just introducing you to you the problem, like the challenges and aims of actually using machine learning methods in this regime of personalized medicine, and in particular to predict personalized treatment outcomes of patients. And we are going to talk about what type of neural network architectures can be used in order to do so. In particular, how we can use static optimal transport as a good inductive bias in order to reconstruct molecular responses in biomedicine. So there will be a more theory-heavy talk, but we keep it light. But also, I think what would be nice to somewhat understand how this phrases or how this basically goes into a dynamic OT that really connects theoretically diffusion models, flow matching, and many things you might have heard of in the past year, and how we can use this then to really continuously study those patient responses to treatments and so on and so forth. And I'm going to showcase one application in doing so, in particular, how this is making impact in molecular medicine. And then we tune out looking at somewhat very nice results we currently are gathering from using those type of methods on an observational clinical cohort study. This is running Swiss-wide in Switzerland, where we are able to predict personalized treatment outcomes of melanoma patients. So let's get started. Let's start with the very first part of what is the problem. So I guess, given that this is a machine learning for drug discovery, somewhat summer school, you're all aware how complex biology is. In particular, the number of cells we have in our body, like on average, 3.7 to the power of 10 to the power of 13 cells, right? And every human is very unique. So this is a very unique and complex biological mosaic. And this complexity does not only arise through scale, but also through the dynamic interplay of those biological processes on the molecular level. This you might have heard in the past days, but also on the cellular level. And here we take much more the perspective on somewhat how can we understand biology on the level of the cell. And then, of course, components of the cell are not only the genes that are driving its function, but also this entire process of transforming DNA into RNA into proteins that then execute functions. This entire regulatory mechanism that somewhat decides which cell type and cell state we are dealing with. So those cells, which even though they are all based on the same set of DNA, take various different functions and forms. And in particular, this is the kind of phenotypic readout we are going to deal with mostly. Every cell exhibits a very, very specific expression of genes, like what genes are active, what genes are not active, and how this is going to basically decide on the function of a cell. And disturbances in this very finely regulated systems are often the root and cause of or starting point of a disease. So I have maybe a mutation in my DNA or something that has been wrongly transcribed into RNA on my protein level, or something even more complicated that is disturbing this system and ultimately changes from a healthy cell state into a disease cell state. And this is, of course, a very coarse and very crude approximation of what is happening here. But what we want to study in this talk is how patients respond to a treatment, and here respond to a treatment by studying how their cellular responses, if I'm able to measure really the cellular profiles, the phenotype of profiles of cells, how are they going to respond to a particular treatment? So I select a treatment as a clinician, I'm giving it to my patient, and now I'm trying to understand how those cells of a patient are going to respond to my treatment over time. And in particular, this is important because I want to understand, are these cells resistant to the drug I'm giving? Are they basically sensitive, and so on and so forth? And we want to learn this with machine learning because the moment a new patient comes into the clinic, we want to make a wise and somehow informed decision of what is the best drug to now give to this new patient with a slightly different molecular profile. Here, molecular profile, again, measured through the set of single cells. And so this is really the kind of root or like core principle of personalized medicine. We want to have patients receiving a targeted therapies tailored to their molecular profile. So how do we capture the molecular profile? I already hinted at this. So by now, if I have, for example, a cancer patient, as this is going to be the somewhat study object in the observational clinical cohort study I'm going to showcase you at the very end of my talk, we can really understand and look at the tissue by basically dissecting the tumor and understanding the spatial architecture, like having a tissue slice. But we can do much more. We can really look deep down at the individual cell level and understand cellular microenvironments. And in particular, of every individual cell, we are able to measure genomic features, transcriptomic features, proteomic, epigenomics. And we can also, of course, record those with like imaging technologies and so on and so forth. Ultimately, what we are provided with is very high throughput data. So really a revolution in biotechnology that has now provided us with high throughput data of thousands and thousands of cells. I'm able to measure and record thousands and thousands of features. And just to give you an idea of what those somewhat like data sets by now look like, I mean, the Human Cell Atlas, for example, has more than 10 to the power of 8 profiled cells. If you look at other data platforms such as CellXGene, you have almost 36 million cells. And this will also be relevant towards the end when I will give you an outlook into what these current works are currently moving to. So on the other hand, right, even though we have this massive amount of data, it's not that our AI methods right now really excel in somewhat capturing well and generalizing across this biological complexity. What we've seen on the other hand is that in fields where AI has really gotten us through several breakthroughs by now, that we have very specific architectures or architectural principles designed for the type of data we are facing with. So RNNs, self-attentions, CNNs, when dealing with language sequential data or images. And similarly, right, we've seen, of course, quite a kind of input and insight by the network architectures, transformers, both for language and vision, as well as the type of loss functions we are using, unsupervised, self-supervised, via masking, next token predictions. And all those mechanisms are, of course, based on somewhat the structure of the data we are dealing with. And we need to find something similar for the biological data we are working with. So we need AI methods, AI architectures that are very much tailored to the biological data we are working with. And biology is much more complex than anything we've seen so far. And here I'm going to highlight mostly challenges out of the single cell biology, so the type of data we are working with. So on the one hand, all the biological measurements we make, they are always indirect, obscured, and noisy. So if I want to measure gene expression, it's not that I'm actually measuring, or gene activity, it's not that I'm directly measuring gene activity often. I'm doing this via basically measuring which mRNA is present in the cell, right? So this is an indirect measurement. Then of course, if I take measurements across different days, across different samples, I always have to deal with a lot of batch effects. So they are very distinct noise sources we need to take care of. Then biology is highly multimodal. There's many different modalities, many different type of measurements, and so on and so forth. Just to give you an example, one of the most recent technologies tries to actually record some of these tissues through multiple layers. On the one hand, we just have an H&E staining, which gives us an idea of the pathology of the overall tissue structure. But then I can also look at imaging modalities that give me an insight into certain locations of proteins and so on and so forth. And then I can measure spatial transcriptomics, so which genes are active and whatnot. So these are very rich, very high dimensional, and very complex data sources we are dealing with. And then last but not least, all the biological processes we are studying, they are inherently dynamic and they are driven by countless interactions. Looking at this from a static viewpoint is not getting us far. And then those measurements are destructive, right? Every time I'm actually conducting a measurement, I need to destroy the cell I'm measuring. But if I want to measure and record and forecast how a dynamic process evolves over time, ideally I would like to measure the same cell several times, right? In order to understand how they change over time. But every time I measure a cell, I destroy it. So I only have somewhat snapshot data, and this core principle of machine learning of having a paired data of the state and what I want to predict, so X and F of X, this pairing is not available here in single cell biology. So we need to already from the root on think about different principles in order to learn from those type of data. So I have non-time resolved measurements like snapshot data, where I sample cells, I measure them, but then I destroy them. And at the next point in time, when I conduct another measurement, it's going to be cells from the same type of process, right? But different cells. So this is why we have basically non-aligned, non-time resolved or snapshot measurements available to study these dynamical processes over time. And so what we want though, to be really successful and excel in, for example, forecasting treatment outcomes, is to have machine learning models that really in silico understand cell behavior. Meaning that if I have a cell which lies in my very high dimensional gene expression space, for example, because I measured the 1,500 highly variable genes, right, that somewhat decide on how the cell looks, or like somehow give me an insight on how the cell looks like. I want to really understand for an individual cell how the cell is going to, for example, transform from a healthy into a disease state. And what does it mean? So I want to really understand the pattern of the change in expression of all those features I measure upon this transition. So what has changed and how has the cell's phenotype changed? Similarly, right, if I give a drug, I want to be able to predict on the level of every cell how a cell has changed. And so here, what I want you to remember, because this is going to be important throughout the talk, is that we have basically a start state before an unperturbed state of the cell. We have the prediction of the treated state, which tells me how the cell looks like after treatment. But then we also have this response pattern here. And this is in particular, this will turn out to be very important in order to understand somewhat the clinical success of such treatments that allows us then to make somewhat decisions on what treatment to select for a particular patient. So why do we want to do this on the level of individual cells? It's because cells are very, very heterogeneous. So a tumor is very heterogeneous. It has lots of different type of cells, and they might very differently respond to a treatment. And as a clinician or as a biologist, I want to understand what are the subpopulations that are going to respond to a cell, to a treatment, and what are those that are resistant, because based on this, I can make my decisions on which treatment to choose. And last but not least, right, we need to have machine learning models that learn from underlying data. as I mentioned already, these technologies are destructive. I don't have an alignment. I only have samples of a distribution that changes over time. So how do we do this? And I'm going to basically introduce a first class now of models that use optimal transport as an inductive bias, and that allows us to reconstruct those molecular responses of single cells to a treatment or whatever type of perturbation you're studying. So let's go through the problem setup again. So I have, for example, a tumor sample of a patient. I'm able to measure these cells through some measurement technology, such as single cell RNA sequencing, that gives me an idea about the somehow phenotypic state of my cell population. This is a sample of cells from the distribution of unperturbed from the unperturbed patient, so the patient before treatment has been given. And now I would like to forecast how the cells are going to respond to the treatment. Basically, how does my distribution of unperturbed states evolve into a distribution of perturbed states upon application of a drug? Again, measuring a cell requires destroying it. I cannot measure the same cell before and after treatment. So the easiest way of dealing with this is, of course, just saying, okay, I'm just going to model the average. The average, how the cell population looked before and how the cell population looked after treatment. Again, this does not at all capture heterogeneous states. I'm not able to say, hey, the blue population is actually going to respond to a treatment, while the gray one is going to be completely resistant, right? I will have no idea about these type of things. So instead, we somewhat need to come up or reconstruct the underlying somewhat alignment or dynamics that steer or that describe the map that would explain the transformation of my distribution mu into my distribution mu. So I need to find a map or somewhat an alignment that transforms my x into the perturbed state T of x. And what is the optimal somewhat map I can find, right? The optimal alignment. Here we need to somewhat make a prior assumption, because there's of course like thousands, like there's an infinite amount of different maps that can potentially describe the transformation of mu into nu. And we are going to make an hypothesis that the cells are all continuously going to change upon drug, like the features are continuously changing, and that the matching that overall minimizes the discrepancy of my cell state before and after. So I'm going to find those which somewhat would follow the path of minimum effort, of shortest path in the space of measure, that this is going to be the most likely map that explains these cellular dynamics, given, of course, some choice of cost function. And with this, I'm basically looking for now a map that somewhat conserves the mass. So I'm not losing any cells. So basically the mass is conserved from before and after, that overall minimizes, right, the transportation cost. And this coincides with a very famous principle of optimal transport that we are going to use as an inductive bias to parameterize those maps with neural networks. And we are not the first ones to do so. So there has been previous work in single cell biology from the Broad Institute that has used OT as an inductive bias for those processes. And this is also something we are going to explore throughout the talk. The power of optimal transport is not only that it serves as a good inductive bias, but also its very close connections to PDE theory as well as SDE theory, here connecting to something that many of you might have heard, diffusion models. And we are going to briefly explore, like on a very somewhat highway, the somewhat connections of optimal transport to these more dynamic somewhat notations of processes that are basically parameterized as PDEs or SDEs. And I'm going to talk about two works, Celot, as well as SB-Align, that's a flow matching method. If you are interested in more, I gave jointly with Marco, my PhD advisor, a tutorial at ICML last year, where we talked much more in depth about the principles of optimal transport, as well as its connections to PDEs and control theory. There's also going to be this summer a Nature Reviews Methods primer that goes through similar the background of, or like somewhat many derivations of OT and how it can be used and its connections, with the core focus on how this can be used in single cell biology. So I will be posting the moment this is online. So what do we want to do now? We want to somewhat parameterize this map such that it corresponds to a map that we would receive when solving an optimal transport. And we want to do so with a neural network. So then if a new patient comes in, we can use somewhat, or we can predict for unseen cells, for new cells, how they would potentially respond to a treatment we apply. The map I showed you before, this optimal transport map, is something known as a Monge map. I had the person on the slide just 2 slides before. Gaspar Monge is somewhat thought about this topic already in 1781. He was a friend of Napoleon, so it's a fairly old theory that is very much rooted in the field of mathematics. And we are going to just utilize or somewhat use certain theoretical properties. So for example, this map exists under fairly general conditions. And in particular, there's a famous result by Bunne from 1987, which states that there must exist a convex potential size such that this T can be basically parameterized as the gradient of this convex function. So we can transform the problem I had on the previous slide into its semi-dual formulation, where now the optimal transport problem is basically phrased via this convex potential and its convex conjugate. So this is the convex conjugate. And what we are going to do, and I'm not going to dive into details here, but we can parameterize this convex potential via a class of neural network that parameterizes the space of convex functions, so-called input convex neural networks. And this is only one way of doing so. There has been by now many, many different ways. And this is already a list that is probably outdated on how to actually parameterize these neural optimal transport methods. So we can, because solving this convex conjugate is quite tricky, we can basically just approximate it via another convex function. And we derive a min-max or max-min formulation that is very similar to those Wasserstein-GANs that have been published now many, many years ago, 2017, where the problem was similar, but the cost function was a different one. It was an L1 cost function, ultimately resulting in Lipschitz constraints. Now here we have these convexity constraints that we can utilize and parameterize, basically constrain the class of neural networks you're looking for, by parameterizing them as input convex neural networks. And so now what we have is basically a parameterization of this Monge map that maps our unperturbed states into their perturbed counterparts. And so this is the core methodology we use in Sellout and Nature Method, the Nature Methods publication from last year. So there is also, right, this is a fairly static formulation that's not very informative of the continuous dynamic process of those cells, potentially, because I only like map my unperturbed states into their perturbed counterparts. But what is interesting is that optimal transport very easily connects us to powerful architectures such as diffusion models and flow matching. And in the next couple of minutes, I just want to provide you an intuition on how this is the case. So far we had this static OT, right? I'm mapping my cells from its unperturbed into their perturbed distribution by parameterizing this through a neural network. But of course, sometimes I'm actually able to observe a cell population at several points in time. And I want to really understand the continuous time dynamics. So we need to somewhat add this time variable into the equation. And we can do so by parameterizing this with PDEs. I'm not going to talk about this today, but also through SDEs. And here we have these nice connections to score-based generative models, diffusion models, which we are going to explore. How does this sudden shift, or where do the dynamics are coming from? In fact, the Brenier theorem we explored on the slide before already gives the first intuition on how this can actually be a continuous time process. Because through by applying a change of variable formula, we actually derive this Monge-Ampere partial differential equation that describes now the continuous dynamics of my measure or my distribution mu0 at time point 0 to a distribution mu1, before we called it nu, at time point 1. And so basically we follow along this convex potential. This has been so far mostly used to study certain regulatory processes or kind of properties of optimal transport. In particular, last year's Abel Prize, as well as the Fields Medal, went to people who studied these type of processes or these type of PDEs in connection with optimal transport, among others, of course. This is not a very practical formulation though, because it's very, very hard to solve and very hard to parameterize. So instead, Benamou and Brenier took a different perspective on the problem and said, okay, so let me study the continuous evolution of my measure mu in time by here parameterized time between 0 and 1, by basically finding a time varying vector field that describes the overall evolution of my measure over time by finding basically the path, the curve, and the space of measure that describes this dynamical system, this underlying dynamical system, the evolution of my measure over time. And of course, we are still in the optimal transport setup, meaning we can't just, we have these mass conservation constraints I mentioned earlier when introducing the Monge problem, that we can't just lose a cell over time, right? We need to somewhat really model the continuous dynamics of every particle. So we need to follow this continuity equation, conservation of mass principle, such that my distribution at time point 0 corresponds to the marginal I measure at time point 0, and the same being the case for the time point 1, really coinciding with the marginal or my measurement at time point 1. And because we also want to look at the optimal transport, right, we still have some formulation of optimality. Before this was via the cost function. Now I'm looking basically for the shortest path. Here described via the length of the vector field. So I want to now look for my path in space that corresponds to the shortest or the minimum path, given the time variable, like a parameterization of my time varying vector field, as well as basically my start and my endpoints, mu0 and mu1. And I'm not going to dive into, but so these type of principles have been actually used to somewhat connect optimal transport to neural ordinary differential equations, as well as connections to flow matching. But we are going to derive it via a different route. So the overall Benamou-Brenier formulation is now, I'm looking basically for this somewhat vector field, V, and the path, minimal path, such that basically I conserve my mass, I have my marginals at mu0 and mu1, and I'm following the minimum kinetic energy. So somewhat a principle fairly well known from a physics, or like kind of that has very much a physics interpretation. We're also going to re-study this problem from a different point of view, again with the physics somewhat interpretation of the process, as these are processes that have actually been used to understand certain particle dynamics in physics. What is interesting though is that we don't need to stop here. So this was the Benamou-Brenier principle, right? We can also look at this from a stochastic control or from a control perspective. So here it was the minimum kinetic energy in fluid dynamics, but much more popular, somewhat this is a very popular formulation that also always appears in control. And now instead of having this vector field, I'm instead looking at my feedback control. So given a closed loop system with some input control and output, which I want to feed back into the system, I'm now looking on how to understand or how to steer the system with a minimum cost. Here, so here I again have my notion of cost. And now I'm looking for the vector field that overall basically constrains or satisfies my mass conservation constraints, that I'm basically coinciding, my state distribution coincides with marginal mu0 at time point 0, and my state distribution x1 coincides with the marginal mu1 at time point 1. And instead of having a continuity equation, we now look at the state distribution over time. I can also add a bit of noise, so transform this into a stochastic diffusion process, as this has very nice properties and certain benefits. So here just adding a standard Wiener process. And instead of now looking at a matching between 2 points, I look now at a time-resolved matching, right? I'm not only looking at an alignment between time point, like this point x and y, but I'm really looking at a time-resolved alignment, so over time from t0 to t1. And the more noise I add, the more noisy this basically path or this alignment becomes. This formulation also, just a quick excursus, actually pops up or is known as a Schrödinger's bridge. And this is because Erwin Schrödinger studied these processes in 1931, 1932 by going through a Gedankenexperiment on what could be the most likely evolution of 2 point clouds of diffusive particles. And so basically now we want to find the stochastic process defined on 0 and 1 that connects my marginals, mu0 and mu1, given some prior knowledge on how the underlying process could look like. So here, this could be something like Brownian motion. And it has somewhat a similar notion as the assumption which type of cost function I'm going to use, which we had in the earlier example of optimal transport. Now I'm looking basically for the stochastic process that minimizes the overall relative entropy, given some understanding or given some prior on the reference process, here mostly chosen a simple Brownian motion. How would this look like, right? My state distribution, if they would follow the Brownian motion, I just have like random, basically, movements around the start distribution. For example, here in dark blue, you see an example trajectory. So of course, we can transform this into some more complicated SDE classes of SDEs that really contain a drift and a diffusion, as this is much easier or like better foundation to actually parameterize the system that describes cellular dynamics over time. So for example, we can just use a drift, a vertical drift with a certain level of diffusivity. And so here now my particles evolve over time just to the right. What is nice is that the solution of the Schrödinger bridge, what I showed you as on the slides before, can be expressed to 2 SDEs that are coupled and that are parameterized through 2 policies, my forward and my backward policy. And these have this phi inside, which is the solution of a system of coupled partial differential equations. We don't need to understand this here, just to give you an idea of where this is coming from. And they are in particular coupled through their boundary conditions. But what is important to keep in mind, the overall solution of the system I showed you before can be expressed by a system of a forward and a reverse SDE, meaning I'm looking at my process starting at mu1, I sample points from the distribution of cells in the unperturbed state, right? And now I'm following my forward policy that leads me or guides me to my distribution of perturbed states. And then I can do the reverse. I can sample points at time point 1. And this backward policy guides me back to my starting point. And this might already ring quite some bells on where this principle has been very successfully reused. And this is in particular when we look at these very powerful generative models that have been appearing in the last couple of years. So here, one of my distributions is always a Gaussian, right? Mu1 might be just a simple Gaussian, where I sample from, potentially given some condition. And I did this just as a test recently, when I condition on a picture of me, EPFL, and a text prompt of generating my future research lab. This is what it came up with. I mean, so A, academia usually doesn't make you more attractive and young, and B, females are also very welcome in my research group, but otherwise they produce very impressive, high quality samples that follow the data distribution of interest. And so another name for this diffusion models is score-based generative models, where one of my SDEs just contains a very simple linear non-learnable F, because I just need to transform continuously over time my data distribution by adding noise until I derive something completely random. Now I can reverse the process and I can start from somewhat random sample, but re-derive the data distribution, or basically arrive at samples that resemble the data distribution or follow the data distribution. So I have a very simple forward SDE with a simple drift, which I don't need to learn, but the reverse SDE is going to contain this so-called score function, which is the marginal density of my forward diffusion at time point t, that we can parameterize. We can't really solve this in closed form, but we can parameterize this with a neural network and use a tractable objective to parameterize and learn the system of two coupled SDEs. In the setup before, of course, we were more interested in generating cell states, right, of a perturbation. And in particular, we didn't have this goal of just generating a random set of cells, but instead we would like to understand the evolution of my cells over time from unperturbed into perturbed, or vice versa. So, of course, a linear non-learnable F is not getting me from a distribution of cells at time point 0 to a distribution of cells at time point 1. This distribution is not easy to sample anymore, and I also don't have a tractable objective. Because I somewhat need to redefine here this drift, the simple F, which has been used in score-based generative models, and needs to be a non-linear drift that really explains me those cellular dynamics over time. And what I derived from the Schrödinger bridge theory just before is, right, we can use this system, which we've seen on one of the earlier slides, plug it into both, slightly simplify, and we rederive the system of forward and backward SDE with a forward and a backward policy, or forward and backward drift, that we can now parameterize with two different neural networks. So different than score-based generative models, we not only have one neural network for the backward process, but we actually have also another neural network parameterization for the forward process. And because this is quite hard to parameterize, it works, and it has been also successfully used to actually generate high quality, or like the interpolation between complicated distribution, but it's still very unstable to learn the system of two somewhat neural networks in a forward-backward fashion, which requires some alternate optimization procedures that are rather unstable. So I'm going to walk you through one of our recent works that uses some insights from biotechnology to derive a slightly easier system, or like basically a diffusion process, like a diffusion Schrödinger process that is easier to parameterize, easier to optimize. And in particular also connects nicely to this idea of flow matching that has been reappearing or appearing through various ways in the last year in the machine learning community. So, of course, right, I showed you that, I demonstrated that we want to learn the stochastic process that describes my cellular dynamics over time. And I told you that, of course, these measurements are destructive. But of course, biologists are very well aware that this is a huge limitation of those technologies. And so there's a couple of recent developments that try to overcome this limitation. One, which only extracts a bit of the cytoplasm and conducts basically measurements based on those. And I can do this several times for the same cell. Of course, the same cell would suffer because it always gets a bit of water removed, right? So what other somewhat like changes in the cellular phenotype this causes, we don't know. But there's also strategies that tag each cell with a genetic barcode that can be read out. And in particular, when a cell divides, it's going to be passed to the daughter cells. Meaning if I destroy the cell because I conduct a measurement, but I find the same barcode, gene A barcode again at a later point in time, then I know clonally it's coming at least from the same cell. It's not the same cell, but at least it's a clonally similar cell. And this is why we call this partial alignments, because such methods like live-seq is very low throughput. We only can sample a few hundreds and not thousands and thousands of cells. And of course, those barcode-based methods, they are very much affected by stochastic cell division and by sampling procedures. But it at least provides us with some information on how those processes might be aligned. And we are going to now learn the stochastic process by finding this, like using the Schrödinger bridge principle. So parameterize this again through a system of forward and backward SDEs. And we are going to derive an optimization procedure of solving those forward-backward policies that is much more robust by using 2-decade-old ideas. One is called Dupes H transform, and the other idea is just coming from classical Schrödinger bridge theory. So again, here, this is my Schrödinger bridge, just here via the Kullback-Leibler formulation. And we know that we can represent the stochastic process now with this SDE, forward SDE. So here there's a slight abuse of notation. I now switch to B instead of F. But we don't know the drift. This is the forward policy we want to learn. But given that we have now partial alignments, we know using Dupes H transform that an SDE condition to start at x0 and that ends at x1, so where we basically see the same barcode again, is given by basically the same SDE as before, just expanding it here with Dupes H transform, which is this conditioning of given we start at x1, that we at x0, that we end at x1. Similarly, for the reference process, right, I can use classical Schrödinger bridge theory that states that basically a Brownian motion process that is conditioned to start at x1 and end at x1, start at x0 and end at x1, can be written through an SDE that's known also as Brownian bridge, that has a drift that we can basically compute in closed form. So now instead of basically parameterizing these two neural networks and solving the system via alternate optimization to have good parameterizations of both the forward and the backward policy, I can simply learn this drift here that describes my cellular dynamics by regressing it to the drift of my reference process. So what I know about the process given the pairs I have available in my data. And so this formulation here, this regression-based formulation, is very similar to what we see in these score matching objectives of these score-based diffusion models, which I had on an earlier slide, where we basically regress against the score that we can compute using this tractable objective I had on the slide. So now we can use this to really study this continuous time evolution of my cells. And it allows us to really understand how, at the very beginning, undifferentiated cell population really evolves into fairly heterogeneous cell states, so with various different cell types. So how is this useful? So why did I walk you through both the static as well as the dynamic formulation of OT? This seems, for like probably many of you, fairly theoretical, not at all practical. I'm going to show you that this is actually a very active component of a current observational clinical cohort study, so that these inductive biases we are using in the static OT part and these really powerful connections we have to diffusion models and flow matching models actually matter in predicting very accurately personalized treatment outcomes of melanoma patients. So what we have here is a cohort consisting of, I think, up to 37 patients, so that's not a lot. But of course, of every patient, we are able to measure thousands and thousands of cells. And for most of those cells, we have their responses to 34 different drugs, because we took these patient cells and basically ran a drug screening, how they would respond to 34 different drugs that are used quite actively in the clinical setting. So these are all approved drugs used in cancer therapy. And so now we want to use part of this cohort to learn a generalized perturbation model, such that if we see a new incoming patient, we can take their cells before the treatment has been applied and use those different drugs to predict for every different drug how they would be responding, and then ultimately what would be the best drug to choose. So we can not only forecast the prediction, but we can also then make a decision of what is the best treatment. And so this is all part of this tumor profiler consortium which is running already much longer than I do my PhD. So this is, I think, already like 5-6 years. I don't know how long. Clearly before I started my PhD. And we are going to use basically data from 37 patients with 34 drugs profiled. And so this is a very heterogeneous clinical study. So meaning these are here my 37 different patients. They are all very, very different. So they have a different number of treatments already previously. They have very different type of driver mutations, and all those clinical meta information, of course, very much matter and are going to determine their response to the treatment. Again, so here we use again this principle of being able to forecast how they are going to respond to the treatment. And so we use basically neural optimal transport methods to do so, so that if a new patient comes, we are able to forecast how they are going to respond. And so now we want to do this, of course, for individual cells. So you have my cell profiled, and now I'm going to predict how the cell profile looks with all the different features I've measured after treatment. And what you see here on the slides is that you have the basically just for 2 different features. I mean, of course, we have much more. We have up to 50 features. And if you use gene expression data, then you have 1,500. Here just highlighted for 2 different features. This is the distribution over the cell states before treatment. And what we want to learn is the transformation of this light blue curve into the dark blue curve, of course, for all features at once. So not only those 2, but also the 50 others. And what you can see in red is the predictions of our method, and in gray, the predictions actually of other baselines that are mostly autoencoder based. And what you can see is that Cellot is very, very much able to really capture this response. For example, here, that is just some cells that are showing an increased level of SOX9. Here, this is also very interesting because this is a bimodal distribution where clearly only one part of the cell population is responding, and we see a shift of this part of the cell population that is showing a reduction in p-AKT signal, while the other population is fairly static. And so this is just here for 2 different patients, so Mukadop and Mogulip, for 2 different type of drugs. We also, and this is something I didn't talk about today, we actually developed neural optimal transport method that can be conditioned on those patient meta information, because of course they're going to determine the response of a patient to a drug. And we can see that it's very important because sometimes for patients with a particular driver mutation, for example, there's going to be methods that are not conditioned on those meta information, completely failing to see that those patients actually exhibit a strong response, right? So here you see basically Cellot that has been not conditioned and Cellot that has been conditioned using this clinical meta information. So it's much closer to the basically target or the treated population, which we measure. And similarly, it can also show that those sometimes like patients with a particular pattern of meta information are not going to be responsive. So Cellot would predict a response while we actually do not see a response at all. And what is very exciting about this is that what particularly matters is the response we are able to extract, right? So by now having a pairing between my cell and its perturbed state, I can subtract those features and really get the response pattern that captures the perturbation effect for each particular feature. And this is really what captures information that is important to use in the clinic, that A, gives us an idea of the overall response, like how much is the patient able to basically survive upon treatment. So here we see the progression, we have survival data of the patient. So we know those who basically died immediately after the treatment, those who survived for much longer, or those who are more or less still alive, at least for a very, very long time. It's in particular the response pattern that gives us insights into how a patient is going to, like how successful a treatment is for a particular patient. So not the state of the disease before treatment, not the state of the disease after treatment, but in particular how they are going to respond, like how successful is the treatment attacking this type of the type of tumor they are dealing with. What is also nice is that we can do a lot of like downstream analysis, right? So given the meta information, the predicted response really captures very distinct subgroups. So for example, those patients that show a particular driver mutation, they're going to overall respond very different than the rest of the cohort. And we can now use this to really study like new clinical associations that give us insights into how certain patients with certain driver mutations are going to respond to a particular treatment. And we get much more of those clinical associations from the predicted responses rather than the predictions on how they are going to basically look like, how the cell states look like after treatment. And so just to highlight one of them, for example, this combination of a drug called paclitaxel and patients that have an NRAS driver mutation, it is very well known in the literature, in fact, that there's a connection between the two because paclitaxel enhances like microtubule stability, which causes cell cycle arrest and cell death, and that patients with this type of driver mutation actually show an activation of a certain pathway that then very much determines the response to this type of drug. And so what we are currently doing, so this is a manuscript in its final days of being written up, but what we are currently doing is really analyzing all those somewhat clinical associations if there's something of interest for this particular type of melanoma cohort. So where are we moving now, right? I already mentioned to you at the very beginning, and so this is work which I'm currently doing my postdoc, and also what we are starting in the new position at EPFL. I told you already that we are now dealing actually with data sets that are massive, right? So we have 30 million recorded cells. Depending on where you are, this is also much more, right? Like pharmaceutical companies, of course, sit on much more data. But most of the type of methods I showed you, they very much take a data set and then they develop a method or they apply a method to it. But they don't look at this in the context of all data ever recorded, right? And here we are moving towards basically biology or towards a GPT era biology that looks at foundation models really trained on all those data that then try to map in basically new data types or the data types of my current study and look at them in the context of basically other recorded data. It gives us much more information on the biology, the underlying biology, than simply studying a problem from a single data set type. So one way of doing so is by somewhat learning a foundation model that represents or has a good representation of a cell state, meaning something people can call universal cell representation or whatever name you give it. But I'm able to basically have a representation of my state of my cell in terms of certain modalities, in terms of certain type of measurements, transcriptomics, proteomics, and so on and so forth. And now we are going to use those universal cell representations in order to do downstream analysis on cellular dynamics, responses, and so on and so forth. And so this means, and this basically enables us to do a lot of different things. So on the one hand, I can potentially in this universal cell representation integrate lots of different data, both microscopy, RNA-seq, ATAC-seq, so all data that so far have been very hard to combine and very hard to basically compare, even though they study the same type of biological mechanism or the same cellular processes. But it also allows me to map in data from different tissues, from different species, and so on and so forth. And then this allows me to potentially discover novel cell states, or if I have a particular treatment, really like states of cells that are very unique to a particular treatment that are interesting to further study in certain downstream analysis, but also to study, this is what we've seen in this talk, how continuous time changes affect cellular states, either naturally occurring or in response to certain intrinsic as well as extrinsic perturbations. And then ultimately, of course, this is more things we are looking into right now, is how does this enable us to really design and engineer new cell states that can be then used as potential therapeutics, or how I can basically have my patient represented. Those patient cohorts are always very, very small, right? But so in context of all measurements conducted so far, it might give me very interesting insights into the state of disease of a patient, and basically complemented with all the other data that are available. And so this is what we are currently working on, like something we call a universal perturbation model, as well as vertical tissue representation. So this is work which is a future PhD student of mine has been starting when I was still at ETH, because these are going to be tools that are relevant to support clinical decisions, such as those which are running in the background of the clinical cohort study I was mentioning earlier. And so with this, in case you're interested in this type of research, like why coming to Switzerland and why particular the lab we are starting now in September, is that on the one hand, right, we are at a very critical point in biology where we have a lot of data, whole organisms, cell atlases, and a very rapid data growth. So data doubling every 6 months. At the same time, we need to have core development of architectures that can deal with this type of data. And this is nothing we have right now. So there's still a lot to do. And we are very far away from the somewhat vision I presented to you on the previous 2 slides. To do so, we need a lot of compute power, and this is something Switzerland has invested in. So we have now a supercomputer infrastructure that is launching this summer, where we have over 10,000 GPUs. And you need to, of course, have somewhat countrywide initiatives that are collecting those patient data and that are very curious and interested in connecting it to the progress made in AI. And so with this, I would like to thank my co-authors that have been all part of the methods presented. So in particular, Gabriele Gut at the University Hospital and Lukas Pelkmans at the University of Zurich that have been mainly generating all the data and providing all the clinical insights and so on and so forth. My 2 PhD advisors, of course, Andreas Krause and Marco Couturi, and now my postdoc advisors, Aviv and Jure at Genentech and Stanford, as well as many students and colleagues I have been working with. So the CELOT, as well as the clinical cohort study is also led by Gunnar Retsch at ETH, and I'm working closely with a PhD student of his, Stefan Stark, as well as Valentin Yaping at ETH, Valentin DeepMind. And if this is of interest, visit our lab webpage and reach out to me. And we are excited about all the future work that will be coming in the next month. And I'm very happy to take questions if I'm not completely over time. Thanks a lot.
We have time for 2 questions. If you have a question, raise your hand and I'll bring the mic to you.
Hello, thanks for the great talk. We've heard today that omics data it's multidimensional, but usually it boils down to actually a smaller dimensional manifold usually. And I'm wondering, don't your flow tend to sometimes, model a cell that at some point becomes a sequence of numbers that does not represent a cell after that time, Doesn't
yes.
the flow go into a regime of not real cells? And is that a problem if it happens at all?
Yes, so no, that's a very good remark. So we are actually also studying those flow in a lower dimensional regime. So we first learn a lower dimensional representation, which we study the flow, because these omics data, even though they measure 1,000 or like actually 20,000 features, right? It's often only like a few core components that really like explain how important to study the overall dynamics. And then we basically map back to predict what are the changes in the gene expression pattern and so on and so forth. But clearly, in particular here for this very last part I was talking about, when you want to do this in silico design, right? Like, and you want to generate really novel cell states. We never know if this is a viable, valid biological cell state. And this is why all those type of experiments need to be, if it's really about like designing a new cell state, they need to be done in somewhat connection with like downstream lab work that tests the cell state you are predicted and feeds it back into the system. For the work I showed you on the slides before, this is less relevant because it's more, okay, we are predicting a cell state, but what really matters is predicting the response of individual cells. So it's not that every somewhat cell state which we predict here upon treatment is really going to be individually evaluated, right? So it's not that we look at every, what is this gene, this gene. We are looking at this more still from a overall cell population, overall response. But the moment you enter regimes such as those where you want to engineer new cell states, this always needs to be coupled with like, okay, so now trying to create this new cell state in the lab and then going back, mapping in the data, trying to understand what this means. So this will always be a lab in the loop type of process in order to understand, is this like similar to creating new molecular design, right? Like ultimately we actually need to synthesize this and understand if this is something that actually has those properties. The same thing will need to happen in biology, where we need to create those cell states in the lab, map them back, understand if they actually behave the way we infer. Yeah. But for the analysis on the slides before, it's less crucial that every cell state is basically a true cell state. It's more like the overall response pattern that really matters to do the analysis.
Another question here. You mentioned about using cell profiles to predict the outcome of a drug, for example, on a specific cell type. Do you work with only one cell type per experiment there, or do you
no
more than one cell type together? I'm thinking of the cases where the effect of a drug on a cell is actually dictated by, for example, something that happens on a different kind of cell, in a different tissue in the body.
Yeah, so I basically always make forecasts on the heterogeneous population that contains multiple cell types. Though, if you give me a new cell type, right, in a new tissue and apply the same model, there's no guarantee that this generalizes. And this is in particular something we are currently studying in this context of this universal perturbation model, that ideally, of course, you want to generalize to all type of cells and to all cell states across all tissues, all species. But those models will always be only accurate on the type of cells, on the cell states that has been seen. So there are no generalization guarantees at all to new unseen cell states and cell types. In fact, we know that they don't generalize. So this is also experiments we conduct. Also same, if I now give a new drug I've never seen, there's no out-of-the-box generalization to a new perturbation type. But it is always operating on several cell states, several cell types, and without any annotation of them. So there's no, I don't need to annotate my cell states for the predictions.